right, so we're going to go through incomplete dominance, codominance, and X-linked or sex-linked traits. So with incomplete dominance, this is a blending of traits. So what that means is that if you have a homozygous individual, homozygous dominant, for example, a homozygous recessive individual, when they would have a breeding event and make all heterozygous children, they are going to have a third alternate phenotype. So a classic example of this is on this page with flowers. So for example, if we have a red flower, a red would be homozygous dominant, 2 capital R's. A white flower would be homozygous recessive, or 2 lowercase r's. When they would breed together, they are going to make all heterozygous flowers. That would be capital R, lowercase r, and that would be a pink flower. That pink flower is that incomplete dominance. It is that blending of traits. So if we look at your practice problems that you have, and we have your red alleles and your white alleles, the phenotype for this plant, the red one, would be red. The genotype would be capital R, okay? For the white flower plant, the phenotype would be white. The genotype would be lowercase r. And then when we put those into the Punnett square, our red flower would have capital R, our white flower would have lowercase r. All of the flower babies would be heterozygous, capital R, lowercase r, and they would all be pink. So they would all be that intermediate color. Looking at our next trait of codominance, this is when alleles appear together. So the example that we use is with cattle, where a white cow that would have 2 of the same allele and a red cow that would have 2 of the same allele, if they would breed together, they would make a spotted cow where we can clearly see the red coloration and we can clearly see the white coloration. So that is an example of codominance. So for an example of codominance, just writing it out, if I would have a white cow, so for example, I'm just going to use the letter W for our white cow, and then if I would have a red cow that would have maybe 2 R's, all of the cows would be all RW. So that would be all roan cattle, or that spotted cow, where you can clearly see both alleles. Blood types are an example of this in humans, where you can have 4 different blood types. The positive and negative, if you've ever heard of like A positive or B negative or whatever, that's something separate. We're not going to talk about that. That goes beyond our scope and sequence. But blood types of type A, type B, AB, and O are the blood types in the ABO system for humans. What this means is if you have type A, if you notice there's like these little circle spike things on your blood, that means that they have little name tags that are a protein called immunoglobulin that is type A immunoglobulin. So it has a little name tag that says, hey, we have type A. If you have type B blood, you have little name tags that say, hey, we are type B. They have little name tags that say, we are type B immunoglobulin. If you have type AB blood, you have both name tags. So they have type A and type B. If you are type O, if you notice, there are no little name tags. So there's basically, I like to call it naked blood. In your body, this is actually part of your immune system, where if you have type A blood, you have type B antibodies. So that means if type B blood gets into your system, like if you have a cut, somebody bleeds on you, something like that, we don't want other people's blood getting in us. That is how a lot of diseases are carried. So you have type B antibodies that kills off that invading blood. If you have type B blood, you have type A antibodies. So if type A blood gets in you, then it kills off that type of blood. If we look at type AB, they don't have any antibodies. So type B blood or type A blood could get in you and nothing's going to happen. If we look at type O, they don't have any of those little spike proteins and they have antibodies for both. What this means for blood donation is if you have type O blood, because you don't have any little spikes, you can actually donate blood to type AB, type B, type A, and other type O people. If you are type AB, you can receive blood from type A, type B, or type O because your body doesn't have any of those antibodies. Okay? If we look over here, looking at our different genotypes that we have, if you have type A blood, your genotype would either be IA, so that means immunoglobulin A, I. Okay, so this I doesn't have a little superscript on it, so that means that it's naked or like that type O. Okay, or you could be IA, IA and have type A blood. Same thing for type B. IB with a little I would be type B blood, as well as IB. For type AB, this is the codominant one, where you have IA, IB. That would be type AB blood, where A and B appear together. If you have type O blood, you have two lowercase i's, so two i's with no superscript. So if we are looking at the practice problem that is on your page, we will do this one together. It says, a type AB woman marries a type O man and she gets pregnant with their first child. Perform the cross on the right to predict the chances of each blood type for their first child. So if we have a man with type O blood, his phenotype is type O. His genotype, according to this O, is little I. And the alleles that he have would be little I. If a woman has type AB blood, her phenotype is A, B. Okay? And her genotype would be I with a little a and I with a little superscript B. And those would be her alleles as well. So you would list those down below. Okay? Filling this into the Punnett square, the man's alleles are pretty easy, just two lowercase i's. For the woman's alleles, we have to make sure that this IA travels together as a unit, as well as the IB. So that is all the same thing, even though it's two letters. Okay? So then we are going to go through and fill in the cross. So we would have an IA and a little I in this one. And then I went through and just filled the rest of them in off camera because I'm old and writing with my finger on my keypad is difficult. So we have IB, little I, IA, little I, IB, and little I. Okay? So what this means is these two kids would have type A blood, and these kids would have type B blood. So if the couple has a daughter, what is the chance she will have type A blood? So that is going to be 50%. And then the couple has a son, what are the chances they have type A blood? That is also going to be 50%. So it starts over with each subsequent child. Okay? And the last unusual genetic trait we have is sex-linked traits. So with sex-linked traits, these are typically always going to be on the X chromosome. So just to reiterate, if you are a male, you are going to have XY chromosomes. If you are a female, you have XX chromosomes. So because males only get one copy of an X, and because males have to get that X from their mom, because you have no choice if you're a woman to give an X because you only have X's, Males are inheriting these conditions from their mom's side of the family, and they only need one affected copy to actually have the trait, be it dominant or recessive trait. Females, because they have two X's, it's just like normal dominant recessive. So they would have to have two copies to get a recessive condition, one copy to get a dominant condition. Because of this, it is more likely for males to have sex-linked traits. Some of those sex-linked traits are baldness is actually sex-linked, hemophilia, which is a condition that causes you to not be able to make blood clots, which can be very dangerous. Another one is color blindness. So color blindness is more common in males because it is this X-linked trait. So using that example of color blindness, we are going to fill in this question here. So if we notice, it says color blindness is recessive, so it'd be X with a superscript E. Normal vision be X with a superscript capital E. So if this man is colorblind, his phenotype is colorblind. Okay? His genotype, he would have an X and a Y because he's a male, and his X would have a little e because he is colorblind, and colorblind is this lowercase e, okay? The woman, her phenotype is having normal vision. The genotype, she would have two X's because she is a female, and both of her X's are going to have a capital E. Okay, so if we go in and fill in the Punnett square, all right, so we're going to fill in the Punnett square, so I have them on the outside. Guys, I figured out how to use a text tool. I'm very excited about this. So in this box right here, I would have X with a capital E and X with a lowercase e. I would also have that in the next box, X with a capital E, X with a lowercase e. In this box down here, I would have X with a capital E and a Y. And then I would have X with a capital E and a Y. So if we notice, the Y doesn't have any of those superscripts on it. So if we're looking at these chances, the bottom are boys. So what is the chance that a baby boy is colorblind? That is going to be a 0% chance. The lights just went off in my room. Okay, and a baby girl is going to have a 0% chance of being colorblind and a 50% chance of being a carrier, because both of these are being a carrier.